The Anthropocene is marked by pretty rapid and radical human-induced environmental changes. And one of those types of environmental changes that we focus on in the lab is environmental pollution, where we get toxic chemicals that are released at high concentrations in the environment. And when many creatures encounter these chemicals, they pose a threat an immediate threat to absolute fitness. And when we think about the fitness outcomes of these encounters in nature, we're faced with this, what I call an irony, that the creatures that we want to kill with chemicals, for example, with pest control, are the ones that tend to evolve resistance to those chemicals. But the creatures that we don't intend to kill with those chemicals are the ones that tend to suffer fitness consequences and decline. And part of the reasons for these sort of alternate scenarios have to do with differences in population size, generation time, and the nature of the adaptive challenge that's encountered. Where these creatures up here tend to have very large population sizes, so very large genetic gas tanks. They tend to have very rapid generation times, and they encounter chemicals, usually one or two at a time, and chemicals that are designed with a specific molecular receptor in mind, right? Whereas these creatures tend to have relatively small population sizes, they've got relatively long generation times, and they often encounter mixtures of chemicals, and chemicals that weren't designed to have specific molecular receptors, so chemicals that might have multiple mechanisms of toxicity. So this poses an arguably more complex adaptive challenge than these creatures do when they encounter the chemicals we throw at them. So when you think about how these kinds of creatures might survive this, many of them, when they encounter pollution, are facing immediate population decline. They're encountering lethal levels of chemical. So that for them to evolve, adaptive variation must be accessible. So where can that come from? So the genetic variation that might rescue a population from extreme environmental change could either come from genetic variation that pre-exists in the population, but for these kinds of creatures, remember the population sizes are small, so their genetic gas tanks tend to be a little bit smaller. Could come from mutation, but their generation times are pretty slow or pretty long, and the environment is changing rapidly in the Anthropocene. Okay? It may come from migration, but for many of these creatures, their landscape is fairly fragmented, so migration can be limited. And it may come from introgression, but that often brings some negative genetic baggage with it. So we've been studying the killifish to try and get at how you go from a normally sensitive fish to a wickedly resistant animal as a vertebrate species, encountering complex mixtures in a short amount of time. So we've published work on the Atlantic coast species that's evolved in some Superfund sites. What I'm going to be talking about today is some newer data that we have on the Gulf Coast species that's evolving in really polluted sites. Okay? So here's the Houston area. So we've been collecting populations from densely collected from along a pollution gradient. Okay, very short geographic distance from Galveston Bay, fairly clean sites. As you get further up into the Houston Ship Channel, you get to more and more polluted sites until you get to the extreme upper Houston Ship Channel or really horribly polluted places. Okay, so we collect fish from these sites. We bring them back to the lab for a couple generations and test their relative resistance to the toxicity of a model toxicant. Here it's PCB126. So here we've got increasing doses of PCB126, and we're looking at developmental deformities that are caused by these exposures. And you can see fish collected from the blue sites, the clean sites, are much more sensitive. They're encountering birth defects at much lower doses than if you were collected at an intermediate polluted site, which are the yellow and red. And if you're collected from a super polluted site, you're not blinking at all. Okay? So the level of resistance to the toxic effects of these chemicals is scaled among populations by the degree of pollution. And the more resistant populations, the level of resistance also correlates with the inducibility of cytochrome P450. Cytochrome P450 is a protein that's activated by this signaling pathway, the aryl hydrocarbon receptor signaling pathway. So when a chemical like PCB or PAH gets into the cell, it binds to the aryl hydrocarbon receptor. This is a nuclear transcription factor. It translocates to the nucleus after it's bound with chemical. It forms a heterodimer with ARNT and activates transcription, which includes cytochrome P450, and that activation of this pathway leads to toxicity. So basically, the more resistant you are to the lethal effects of these chemicals, the more desensitized the aryl hydrocarbon receptor signaling pathway is. Okay? So that's part of the mechanism of resistance. So we're asking next, what are the genetic changes that take you from 0 to 60 in such a rapid period of time in these fish? So there we used a genome scan approach, and here we're looking across all the chromosomes, looking for footprints of natural selection and genetic variation, where we're comparing a polluted site population, the super resistant population here, to the two reference site populations from the clean, another, and then the intermediate resistant populations as well, compared to the sensitive populations. Scanning across the genome for signatures of selection, which we're seeing here, these red steeples across this genomic landscape. And as you can see, for most of these populations, we got this sort of, at the same loci, we're seeing these signatures of selection. And that's also true for these intermediate populations. If I blow up the scale here a little bit, it starts to become apparent. If you stand on your left foot, you can see, close one eye, all of these signatures of selection tend to be lining up. Okay? And when we look under the hood there, what are the genes that we find in these outlier regions? Quite conspicuously, we find a lot of the key molecules that regulate the signaling pathway that we know is adaptively desensitized in these resistant animals. So it's a polygenic story, but what I'm going to focus on today, for what I want to communicate today, is what's going on in this outlier region in chromosome 1. This is work by Noah Reed, and what he discovered is a deletion in aryl hydrocarbon receptor. So this deletion cleaves off the last axon of 2A and the first 6 exons of the tandemly arrayed gene aryl hydrocarbon receptor 1A. If we look at the genetic variation that flanks this deletion, okay, so if we look at the genetic variation, the haplotype that carries this deletion, we see that if you carry the deletion, you've got a quite distinct haplotype than if you don't carry that deletion. Okay, these are populations, right, about 20 kilometers away from each other. A big surprise came when Noah put these data together with our Atlantic coast species. And we looked at the haplotypic variation there. And what we see is that this deletion haplotype is much more similar to the Atlantic coast species than it is to populations of the same species 20 kilometers away. So the Atlantic coast species is also, a few populations have also evolved resistance to chemicals. In one of those populations on the East Coast, a deletion at this locus is also swept to high frequency, but it's a different deletion. We've now found at least 5 different deletions at this locus associated with resistance to toxic chemicals in 3 different species of fish. So when we looked at the coalescence time of these different haplotypes, the deletion haplotype and the haplotype of the intact locus, we find that the time since divergence is much, much reduced if you carry the deletion haplotype, and it appears to come from the Atlantic coast species. It at least is much more recent than species divergence time. Okay, so in the words of Noah's email, his next email a couple months later, something crazy's happening and we need to figure this out. So this is where Sivan and Kristen come in, 2 very, very talented theoretical population geneticists. And they developed an extension of their DMC algorithm that was designed to distinguish selection on young adaptive introgression from selection on standing variation. Okay, the acronym being DMC, my Adidas. Yes, it's clever. But I assure you the science is even more clever than the acronyming. And what they did here is they contrasted a bunch of different alternate models, trying to estimate the generation, the time since introgression, and the selection coefficient of this locus. What they find is the introgression is very recent. We're talking 30 some odd generations ago, and the selection coefficient of 0.8. So we have an adaptive haplotype that offers a huge fitness advantage that introgressed recently from a species thousands of kilometers away. So that was this locus here. That's that very recent introgression. We've got evidence of one other region of the genome that came from the Atlantic coast species, and that's on chromosome 10. Conspicuously under the hood there is the dimerization partner for AHR. And at that locus, Savant and Kristen's modeling showed us that the age to introgression was also very, very recent, and with also a massive selection coefficient, 0.55. So here's Houston Ship Channel. This is where all this business is going on. There's the closest population of the Atlantic coast species. That's pretty far away if you're a fat little fish the size of my thumb, right? You didn't swim there. So we speculate that this introgression event was most probably human-assisted in some way. So we come back to this irony here. Here we've got, with the killifish, we've got a species that we would sort of classify with these creatures down here. So what's going on? Well, their genetic gas tank is not the size of most vertebrates. Most vertebrates, their nucleotide diversity is much lower, for example, than arthropods, but killifish tip the scales here for a vertebrate. They've got insect-like levels of nucleotide diversity, so that probably fares them well. Here's a story where adaptation has been polygenic. There are many regions of the genome that are swooping along here, but there's no one single SNP that's swept to fixation. We've got the aryl hydrocarbon receptor signaling pathway that appears to be this really big lever for natural selection. We've got evidence of adaptation from standing genetic variation, where their massive population sizes as a vertebrate animal is probably putting them in a good position to evolve quickly. But that wasn't good enough to be able to evolve and maintain fitness in these most polluted sites. That required at least a couple loci that were contributed from this Atlantic coast species. And clearly this selective benefit offered by these adaptive introgress loci outweigh any negative genetic interactions that might have accompanied those haplotypes. And the arrival of these haplotypes was probably enabled by human-mediated transport. So with that, these are the folks that did all the heavy lifting, particularly Elias, who did the selection scan work and the phenotyping, Noah, who did the deep dive and discovered the deletion and the introgression, and Savant and Kristen that came in and did some amazing modeling work. We're recruiting a postdoc in the lab, and thank you for showing up. Yes?
Do you have any hypotheses as to why this deletion might be adaptive?
Well, if you're a developing embryo and the aryl hydrocarbon receptor pathway is inappropriately activated, you don't develop a heart properly. And that's pretty much lethal by the time you hatch. So if you're able to desensitize that pathway, you turn off that toxicity and you survive. So if you go into a mouse and you knock out aryl hydrocarbon receptor, you can't kill it with dioxin. Yeah?
Well, why the multiple parts of the pathway, why does all have to be deactivated or changed? Or do you
We don't know.
Mode of selection acting on, like, so you have this initial one that desensitizes, and then later on you have these overexpression of these different parts. They can be
Perhaps these are compensatory adaptations that are sort of accompanying it, destabilizing other parts of the pathway that were really screwed up by this, or we don't know. What would be really nice to know is the temporal sequence of when these different components of the pathway were sweeping. We've taken some sediment cores from some of these sites and tried to get killifish DNA from going back in time, but that's had a big success. Yes?
So you're finding your Atlantic populations are grouping with your local populations of the landscape. Do you think are you having genome-wide maintenance of these introgression events across the genome, or is this a focal region that is kind of a special case?
That's a great question. And can we talk afterwards?
Yeah.
The gong is gone.